Welcome to Max Planck Florida's Institute for Neuroscience Neurotransmissions podcast. I'm Jeremy Chang, and today I'm joined by first-time co-host Akash Pal. Akash is a postdoctoral fellow in Dr. Tian's lab. Dr. Tian is a new director at MPFI, and we're really excited to have her. Akash, do you mind telling us a little bit about what the Tian lab is doing and your research interests?
Yes, of course. Happy to. Like Jeremy mentioned, I'm a postdoc in the Tian lab. We just moved here about 4 or 5 months ago from UC Davis. Our lab mostly makes genetically encoded sensors for neurotransmitters and neuromodulators. Our main goal is to provide these sensors for the broader neuroscience communities to help them answer their scientific questions.
I think I speak on behalf of most of the Institute, or all of the Institute, that we're really excited to have you all here and looking forward to seeing the science that you guys are doing.
Happy to be here.
So today we have a special guest with us, Dr. Jerry Chen. Dr. Chen is an associate professor of biology at Boston University and affiliated associate professor in the departments of biomedical engineering and psychological brain sciences, where his lab studies neural mechanisms of perception and abstraction. His lab tackles these questions at the circuit level and also at the level of individual cell types, and has been pioneering novel methods for linking cellular activity to cell types. I'd also like to add in that Dr. Chen is a really special guest at the Institute. He was a postdoc nominated speaker. So every year the Postdoc Association at MPFI gets the chance to nominate a speaker to bring in for a speaker series. And Dr. Chen came in and gave us a wonderful seminar on his science and talked about his career progression. And it has really been, for me, an entertaining day. And so looking at our conversation with Dr. Chen and Akash, what are some of the highlights of the things?
I think some of the highlights would definitely be the fact that we talked about two-photon microscopy, how it has come up through the ages from being something that you have to build in your lab to something that you can just buy. The current generation of genetically encoded tools that have helped neuroscience and neuroscientists. And also the importance of imaging, not just the area of the brain, but also figuring out what specific cell types that there are that lead to these different behaviors. And he also talked about how currently he's in a sabbatical and how it's important to go to new places, get new perspectives.
And so I guess without further ado, we'll throw to our conversation with Dr. Chen. So Dr. Chen, your lab studies mechanisms of perception and abstraction, and particularly you focus on circuits within the cortex. When you talk about perception and abstraction, what do you actually mean by those two terms?
Yeah. So, perception, the way I think about perception is like in terms of what our brain is doing, right? We're trying to understand our environment. Like we're walking through the world, we're looking at new things, we're, touching things, listening to what you're saying, things like that. And so there's two things that our brain is doing, right? We just have to take the physical elements of the environment, color, light, sound. We have to process that information, right? And then we have to extract meaning from that information. And in my mind, the way that we extract meaning is that we have to take our prior experiences, things that we've learned over the course of our lifetime, to then interpret that thing, to know that, I'm looking at your face, I'm looking at your face. These are two different faces. They're not just a bunch of eyes, and glasses and stuff like that. And so there's this kind of dialogue that's happening in the brain somehow where, right, one part of the brain or certain parts of the brain are just extracting the features, the other parts of the brain are trying to, apply meaning and subjectively to that. And abstraction kind of means this idea of like taking this high-dimensional world, right? There's a lot of different things going on, but reducing it into a way that is interpretable, that I'm looking at a face, mm-hmm. It's a concept, mm-hmm. right? There's these ideas. And so on one hand, there is that, when you think abstractly, right, just to kind of by definition, abstractly, there's this idea that things are kind of take more of a kind of a conceptual, semantic meaning, right? But then the brain somehow has to condense that information in order to make it abstract. And that's what people sometimes refer to in like a mathematical sense, that the world is high-dimensional, but yet the brain represents these things in a low-dimensional space. And this kind of compressed format, this abstract format, also has properties that will allow you to engage in the world, meaning that you can infer things that you have not experienced because you have learned one relationship with another relationship, two objects maybe that are not necessarily connected, if you learn these relationships that A is related to B and B is related to C, you can infer that A is related to C, right? And that's kind of this idea of abstraction, that your brain is somehow allowing you to make these inferences through this types of by compressing such information.
Right. So, like in the context of faces, as you were saying, right?
Yeah.
It's the idea that a face is composed of eyes, nose, mouth, but you're not necessarily. You're kind of thinking of it as a whole,
right,
as a face, rather than those individual components, right?
Yes, and or this idea of invariance, right? If you turn your head, if I look at you, from the back of the head versus the front of the head, I still realize, no, it's you, right? There is some kind of invariance to that, to what I'm seeing, right, and perceiving, and that is the idea that these things are being compressed somehow into some sort of abstract representation.
As you're studying perception and abstraction, which regions of the brain are you actually looking at?
So we're looking in a mouse. And so if you think about a mouse in terms of what it cares about and what aspects of the sensory environment it cares about, maybe what people don't realize is that mice use their whiskers to feel the environment, right? So you imagine mice, a lot of times are running around in tunnels and, underground. And so they're not necessarily moving around in areas where there's a lot of light and, they're confined spaces. And so as they're running around, they're brushing their whiskers against things in the environment, right? And so the way they feel their environment is through the whiskers, and they have, a similar amount of tactile acuity in the whiskers as, we do with our fingertips, for example. So it's a great system to study sensory processing in a mouse that really uses their whiskers to feel their environment. And so we're studying parts of the brain that are processing this information. And there are a lot of parts of the brain that process information. You can go from the periphery, right, peripheral nerves. This information converges into the cortex, somatosensory cortex. So we've been studying, somatosensory cortex, but then we're also studying some more higher level areas that we think are involved in kind of, giving more meaning to some of the sensory information that's being processed.
And then when you mention these other higher order areas, what would they be?
Yeah, so I like to make some comparisons to the visual system in primates and humans, right? Because in humans and primates, there's kind of a hierarchy, right? And you have the primary visual cortex, but then information gets passed on to these kind of second higher order visual cortex, and there's kind of hierarchy of information. And as you proceed along this hierarchy, you see that these lower level features get combined into higher level features that give rise to things like face processing, right? In mice, it's unclear whether or not mice are perceiving objects and things like this in the same way that primates are in tactile sense, but there is still a little bit of a hierarchy. There's a little bit of an idea that the somatosensory cortex is processing lower level features, and that features then get sent to higher areas. And one of the higher areas that we're studying is an area called perirhinal cortex, which is an area that's thought to be important for higher level perception. Also relates to learning and memory of sensory information.
Gotcha. And then so you use 2P imaging to look at the
2P being two photon.
Sorry, two photon imaging to look at the perineal cortex. And then you mentioned previously while we were chatting outside of the podcast as that when you first started in 2P, systems were not pre-built. You have to build from scratch. So what has your experience been as the technology has evolved?
Yeah, so I started using two-photon microscopy, which is a way of imaging the brain kind of non-invasively, but with really high resolution. You can image single cells or even subcellular structures, right? It's a very high-resolution technology. This is about 20 years ago when I started to use this microscope. The microscope unto itself as technology had been around maybe even 10 years prior to that. But it was really in the hands of people who were physicists, who really knew how to build the microscopes and use them, right? And I kind of came around, started doing neuroscience around a time where it was starting to transition from people who only knew how to build microscopes to maybe more experimental neuroscientists, biologists. But it still was not quite at the stage where you could buy one commercially from a company and have it built and it was already like a turnkey system. So I kind of got some familiarity with these microscopes and how they're home built, and it's interesting learning these home built systems because you really know all the parts in and out, and you kind of know how they work together to generate images. And there's been a lot of technology development to improve these systems. They can go faster, they can go deeper, and they can image larger parts of the brain and things like that. And so, I myself, doing my PhD in a lab, whose collaborator were pushing the edge of the envelope, so to speak, of this system. And I can appreciate how your ability to be at the forefront of developing new systems enables you to kind of look at parts of the brain in a way that maybe you weren't able to do so before. So it's really an enabler of being able to ask new questions. And so, over the course of, my training, and then I started in my own lab, I always took this kind of idea to heart that, being able to advance these tools and use them really gives you a lot of power. And so that's kind of why, even as there's been technologies available where you can commercially obtain these microscopes, I've always kind of opted to maybe still continue to build them in our lab, and we can have the capacity to tinker around and push and use them in unique ways.
To have the familiarity with the hardware.
Yeah, to have the familiarity, to know kind of where the limits are currently with the current technology, but then also see what's possible. Other groups that are more purely engineering groups or in purely physics labs, they're pushing these technologies as well in different ways, and see what they're doing and recognize how, what they're doing, we can bring in some of these elements into our own lab, combine them, and we apply them in the right way, we can start to look at the brain, and with higher resolution and with higher, temporal resolution, things like that.
Mm-hmm. Right, so I think that brings up an interesting question. What are the sort of areas that developing your own microscope opens to you that and what are the sort of the biologically relevant questions that those areas are able to tackle? Yeah,
so one of the things that we really worked on is being able to understand how different parts of the brain communicate with each other, right? So in our system, our whisker system, mice are really moving their whiskers to feel the environment. So there's a both sensory component, but there's also a motor component. Animals have to move, and there's this dialogue between the sensory cortex and motor cortex that really works to integrate this information. So we really want to be able to capture not just the sensory cortex by itself, but also the motor cortex simultaneously to look at these interactions. Mm-hmm. So that led us to develop ways where we can look across larger parts of the brain and image them simultaneously. Up to now, with an FSU's, past couple of years, maybe there was only the capacity to look at a single part of the brain, one at a time. But now with some of these newer microscopes with larger objectives, larger optics, you can now start to look at the brain over these longer distance, and that again allows us to answer these kind of questions that really, one couldn't have asked, in prior versions of these microscopes.
Mm-hmm. And so then, when you talked about how you're trying to get this sensory information, so what kind of genetically encoded tools are you using to get this information?
Historically, the main means of being able to record activity through imaging approaches is using sensors that report calcium. So the currency in the cell neuron communication tends to be the action potential, right? Electrical activity, but recording electrical activity or imaging has been kind of limited up to a certain point. We've been using calcium as a surrogate because anytime a neuron fires an action potential, then calcium enters the cell. And so you can use sensors that detect the concentration changes in calcium, right? The calcium changes are relatively slow compared to the action potential. So then you're kind of looking at some communication, but it's kind of like a slower kind of communication, right? You really want to look at, the electrical signals directly, right? And so there's been a large push, recent years to measure image voltage more directly. And so through advances through, other research labs that are experts in protein engineering, they've developed these voltage sensors that now demands technologies that requires you to image at the speed of the action potential, which is 1,000 times per second, for example. And so we've been using these voltage sensors, but we've also had to develop other microscopes, right, that will allow us to image at much higher speeds so we can capture now what these sensors are kind of offering to us.
Mm-hmm. And what would you say is your favorite genetically encoded tool?
I think the one that answers the question best, I guess. Yeah. And so for its different questions, it requires different tools. I think the voltage indicators really allow you to ask questions both in terms of with, the millisecond resolution, how networks are talking to each other, but it also allows you to measure what's called subthreshold signals. And so subthreshold signals are kind of the inputs that a neuron is getting before it decides whether or not to fire an action potential, right? So it's getting a lot of inputs from thousands of other neurons that are connection. Each one is causing some electrical activity, but it requires enough of that electrical activity to generate the spike.
Like the all or none principle.
The all or none principle, yeah. And so, but there's a lot of interesting things going on in terms of subthresholds. And there, I think the subthreshold stuff really allows you to understand how a single neuron is computing information, really, because you can really see not only within an entire neuron the subthreshold, but then you can go to specific parts of the neuron, the dendrites or the synapses, and see like each individual compartment, what is it getting, how is that information getting integrated to cause this all or nothing spiking of the neuron. So I think that has a lot of potential to open up doors for deeper understandings in terms of these kind of basic mechanisms for neuron communication integration.
You artfully dodged the question of what your favorite genetic engineered tool is, but if you were to design this imaginary perfect genetically encoded tool for reporting activity in the brain, what are the sort of characteristics that you would want from that?
Yeah. So things that are faithful to the biological process, right? So some of these sensors, they work well, but then there's some questions as to whether or not whether the dynamics of the sensor really reflect the biochemical process that's happening, right? Calcium, for example, there's some questions about whether or not the calcium indicator itself really represents the calcium signals. Mm-hmm. And so I think in general, you want something with high signal to noise. You can really reliably measure the information, and in terms of temporal dynamics, really reflects the molecules that it's meant to detect and things like that. I would say also in terms of tools, there's just an explosion of tools available these days, right? We mentioned calcium indicators, voltage indicators, but there's neuromodulators in the brain, right? That are acting upon a lot of the neurons, and there are a lot of sensors that can detect things like serotonin, dopamine, acetylcholine, right? There's just, in some ways, more sensors available these days than maybe one can spend the time as a single lab to really, investigate and explore.
So you've been using these tools to investigate different aspects of perception and abstraction, but you've also been combining imaging in vivo with classifying cells after the fact. And some of your early work in this was looking at different cell types within barrel cortex. Mm-hmm. Could you tell us a little bit about those two cell types and how that was a sort of conceptual leap in our understanding of circuits within the brain?
Yeah. So the idea is that the neurons and the circuits in our brains are composed of different cell types. You can think of them like circuit components, and these circuit components are organized to potentially carry out certain functions to then for sensory processing, like in barrel cortex. And so we have developed a way where we can measure activity in the brain, but then we can look back and ask, well, what type of cell type were you? Were you an excitatory neuron that caused other neurons to fire? Were you inhibitory neurons that caused other neurons to stop firing? And then we can look at the breakdown, subdivisions of different cell types, excitatory and inhibitory neurons. And one cell type that we identified, which hadn't been identified previously because we didn't know the genetic signatures that compose that cell type. So when we studied it, it is an interesting cell type because it acts kind of what I refer to as a circuit hub for sensory processing. So the idea is that, as I mentioned, when I think about perception, there are circuits that are involved in processing the world around us, and then there's cells that are involved in providing maybe the context, the internal prior information, right? And there's interaction with these two. The cell type that we identified, we think, is one that kind of organizes circuit in situations in which we have a lot of salient information that comes into our system, right? So we might be as we explore our environments, we might be in situations where we have a lot of strong sensory evidence. We have a lot of clear, like sensory cues to understand what we're looking at or what we're feeling. And then there's times in which, we're wandering through an environment that's a little hazy, it's foggy, it's unclear, and we have to kind of fill in the gaps with our prior knowledge, right? And so there presumably are certain cell types that are involved in helping you fill in the gaps, and other cell types that are there to kind of say we have the information that we need. We don't need to fill in the gaps, so to speak. The cell type that we identified, I think, is something that allows us to readily tell the rest of the brain that we have enough information to know what to do with what we're seeing or we're perceiving. We call it like a feature detector, right? So these things are responsive when we have very salient, relevant information, and when we see or experience the relevant information, this neuron goes off, and it tells all the other neurons that, look, there's something information, and there's something interesting going on, but I already know what it is. I don't need the other parts of my brain to tell me what a figure is out. And so it organizes circuits in a way that just provides that information to other areas of the brain that might be needed to use that information for decision making or or for other types of processing.
Mm-hmm. And I understand that because you're interested in specific cell types, that's why currently your workflow, you do the imaging and then you interrogate the same cells that you image to see what kind of cell types that they are. Could you talk a little bit more about that?
Yeah. So cell types, right, are composed or defined by different sets of genes that are being expressed. And in more traditional methods, there's ways to identify neurons by a single gene, but it doesn't necessarily because you can label that neuron with a single gene, but because of the fact that some of these cell types are defined by combinations of genes, these prior technologies don't really lend itself to teasing apart these cell types. So you need other approaches that will allow you to take a neuron and then ask which combination of genes are being expressed into that neuron to allow you to really identify it as, one cell type versus another cell type. And so the approach that we take is one in which we can record from a neuron, and then in fixed tissue and specimens, we can then apply chemistries where we can then stain for multiple genes. And because now we can stain for multiple genes, we can take that information and then say, oh, this is this type of cell type, right? And this is a type, and this is what, versus another type of cell.
You do have a fun acronym for this.
Okay.
do you want to tell us about that?
Yeah, the acronym is called CRACK. Not in a pharmaceutical medicinal way, but we're thinking about cracking circuits, cracking the code, so to speak. And so that's kind of what the acronym stands for, and it means comprehensive readout of activity and cell type markers. So that if you kind of look for the letters, you'll see that it spells crack. Yeah.
At what point do you stop dividing cell types?
Yeah.
Do you have a philosophy for, as you approach that question, as you're looking at these different circuits, when should you stop looking at those cell types? And I guess in that, towards that, does that sort of inform the way that you're choosing how to probe these circuits?
Yeah, so I think there's a lot of questions there, right? So the idea is that cell types, are cell types considered discrete elements that are functionally and anatomically or non-overlapping with other cell types and so forth? Or is it the idea that the neurons in our brain constitute like a continuum, mm-hmm. and that there's a continuum, and this continuum is defined by this gene expression patterns and stuff like that? Based on the evidence that we see, I think the answer is like somewhere in the middle, that there are, by all purposes, some discrete categories that you can break certain neurons into. But at the same time, if you look more closely, what seems like discrete elements actually reflects a continuum, right? So there's that one element, but then the question is really, what is the function of these particular types of neurons? And I think the one thing that, that in terms of cataloging cell types and how that relates to what the biology is, that a lot of times we're picking out genes and we're using those genes to classify the cells, right? Just to give them names, right? But, to the extent that we're actually looking at what the genes are doing in the context of the neuron is not necessarily something that everybody's looking at, right? It's more a way to kind of classify, but, presumably these genes are in the particular cell for a reason, and those in that genes are defining the function of that cell, right? And so I think it's important, in some ways it's handy to delineate neurons into cell types because we can kind of talk about these things in kind of a, in an easier way. Mm-hmm. But on the other hand, I think it's helpful to also recognize that the genes that define the cell types have a function. And so we also need to understand what are the genes doing in these particular cells and how are that contributing to the cell type. So I think some of the things that we're doing is kind of moving that direction where, yes, genes are differently expressed in the given cells, but what are the genes as a program? How does those genes that are defining the cell types actually contributing to the function of the cells in the circuit?
I think in many ways there's kind of a nice parallel here between what we were talking earlier in terms of abstraction, right? These cell types are in a way an abstraction to understand circuits.
Exactly, right. So we're trying to apply meaning, right, to these cell types, these definitions, and we're trying to, and it's a handy way to kind of extract common principles of brain function. And in some circumstances, I think yes, we can extract principles, but on the other hand, we might be missing things, right? If we're kind of not really looking more broadly in a more comprehensive manner of all the genes that are really contributing to these different neurons and their functions.
When answering a scientific question, do you already have an idea which particular cell types you might be interested in, or do you first do your experiment, see what kind of cell types there are, and then go from there?
I think we do a little bit of both, right? So, definitely the field has been working a lot on the cell types. There's already a lot known on certain cell types. And so, on one hand, we want to leverage that prior knowledge in terms of what people have already discovered. On the other hand, I think there's still room to kind of go in an unbiased way where there's hundreds of thousands of genes, like I think we barely scratched the surface on which sets, what genes are contributing. So the experiments that we're doing is a little bit of both. Sometimes we pick up the genes based off of prior evidence that we think it's important for labeling the cells and looking at that. But at the other times, we're interested in also going and kind of asking more broad, unbiased questions like, well, okay, let's just add all the genes, and then let's see how the genes themselves tell us maybe what the functions are and things like that.
What are the core technologies that are actually allowing you to look at the different gene expression profiles of these cell types?
Yeah. So the recent years, there's been this kind of development of what people refer to as spatial transcriptomics, which is a way to be able to read out gene expression across, the whole transcriptome, so to speak, of a cell, but do it in situ, meaning that it's done in the tissue sections. And that really, prior before you had to dissociate the cells into individual cells, and then you run it through a sequencer, and you don't kind of lose that spatial information. But now in spatial transcriptomics, you can maintain that spatial information, and that really lends itself to combining those technologies with imaging approaches like two-photon imaging, where you're imaging neuroactivity. So you see the 3D structure, the 2D structure of the neurons and how they're firing in the context of the tissue, and then you can register that to the spatial transcriptomics technology and really combine the two together.
So let's switch gears a little bit. So you're currently on your sabbatical?
Yeah.
So can you tell us about a little bit what a sabbatical is and what you're doing during this time?
Yeah, so sabbaticals are, if you're in at university and you're faculty, every about 7 years ago, you get the opportunity to take a break from your teaching commitments, maybe your administrative commitments, and it's an opportunity to focus on your research. Maybe it allows you to kind of refresh, maybe explore a new direction, and then kind of come back with a new set of questions, a new set of techniques that then you can then continue on with your research into your next sabbatical. And so for me, I've gone to England to the Wellcome Sanger Institute, which is an institute where they've sequenced half the human genome. It's known for genomics. And, this area of spatial transcriptomics, it's a very fast-moving field. We've started to use some of these technologies, before, but it's moving at a pace where, there's a lot more opportunities to use these systems and technologies than maybe I can appreciate, right? And so for those reasons, I've decided I wanted to go somewhere where I can really immerse myself with other researchers, people who are really focusing on spatial transcriptomics, other type of genomic technologies, to really see firsthand, what they're doing, where are the things that they're working on, where is the future looking like in this technology, so that I can kind of bring that knowledge back to the lab, and we can start to enrich our lab's research program with that, with those new sets of technologies and science. Yeah.
So now that you're away from the lab, do you have any FOMO or fear of missing out? Fear of missing out things happening in the lab?
I stay pretty good in touch with things happening in the lab. I think in the days of Zoom and Slack and remote work and stuff like that, I have a pretty good sense of the science that's happening. Of course, I miss out on the social aspects of being able to hang out and talk to people in the lab, interact, and having that kind of more, casual, interactions. So those are things that I'm looking forward to when I get back. But I think overall, the short-term period of being away, I hope will have long-term benefits in terms of where we can go, and the science that we can do.
Mm-hmm. And then I was actually looking at your lab website, and I was looking at your lab and your lab members, and I see that you all have some data scientists in your lab. So as a neuroscientist, having a data scientist in the lab, can you tell us how that has helped you in your research?
Yeah, these days, right, the types of science experimental work that we're doing, we're generating tons of data, right? More data than we know how to handle and to deal with, right? And it's getting more and more the situation where the proportion of the lab that's doing experimental work versus the proportion of the lab that's doing analysis work is kind of, the balance is starting to kind of change, right? And when you're analyzing data, right, there's a lot of things that one has to do, not just to analyze data, but how to curate the data, store the data, figure out ways to leverage high performance computing to work with the data. And so that really requires somebody who's not just a biologist, but somebody who's really trained in data science to be able to kind of wrangle this data and organize it in a way where we can easily answer the questions and analyze and study the data. Yeah.
And so these data scientists, what are the sort of backgrounds that they typically come in with? Are they biologists by training? Are they computer scientists or physicists? Or?
Yeah, we've had a range of people come in. So we've had pure computer scientists with maybe backgrounds in machine learning, computer vision, with no knowledge of biology that have come in, but with an interest in learning biology, and they've been able to kind of bring in their skill sets and kind of grow and learn biology as a result. We've also had people who are maybe more biologists by training, but have an interest in developing their skills and more computational work, and they've been able to kind of grow in that sense, right? They have the background in biology to really kind of focus their analysis. They know where the more interesting questions are, and at the same time, they're building up their skill sets to, learning the newest programming languages, knowing the newest cloud environments to incorporate. So it's definitely an interdisciplinary. Data scientists themselves, right, are, I think, coming from a lot of different backgrounds and bringing a lot of different skill sets. And I think it's good to have people who are from these different backgrounds. Yeah.
I have another very important question.
Okay.
When it comes to mac and cheese,
Okay.
Kraft or Velveeta?
You have to ask my daughter that, because she's a mac and cheese expert.
Well, thank you, Dr. Chen, for your time. And we're really looking forward to seeing how your time away from the lab will sort of spur on your future research.
Okay, thank you very much.
Thank you very much.
Great, thank you.
Hey listeners, thanks for listening and for supporting the Max Planck Florida's Neurotransmissions podcast.
Behind the scenes, MPFI has a dedicated crew for communicating our outstanding science and engaging with the public, but this is only made possible because of your charitable giving.
Please visit mpfi.org/donate to support our science and our mission.
Thank you.